Welcome to this final presentation, which covers the way that plants defend themselves against pathogens. You will need the worksheet which I've provided for you, which allows you to annotate the diagram in one of the slides on here, and also you can summarise some of the main ways in which plants defend themselves. Quick look at the learning objectives that we've got here. We will be looking at the way in which plants defend themselves against pathogens, the production of chemicals, and the ways in which the plants can limit the spread of the pathogen once the pathogen has got inside. So chemicals like caloes, for example, which I'm sure you probably haven't heard of before. So why are plants so vulnerable to attack in the first place? Well, obviously they're a very rich source of organic molecules and minerals inside the cells, and that would be attractive for all of these groups of organisms that we've got here. And you can see here that a fungal attack on these leaves is causing quite a lot of damage, and particularly these type of leaves which are quite thin, and they're only seasonal leaves, so fungi tend to be more noticeable as the summer goes on. We've got some spines here which give the plants a little bit of protection. That will deter things like insects and vertebrates and so on trying to get into those plants. We've got some bacterial cells here which are shown invading those tissues. Aphids are well known for trying to remove the sugary sap that's found in the phloem. So plants need some ways to try and make them less palatable. Usually they have chemicals which try to deter them, usually quite unsuccessfully. And this just shows some bacteria which have managed to get inside those cells. So obviously plants not only have the organic molecules, but they do form the important foundations for all food chains, being the producers in those. So do plants have an immune system? Well, hopefully all of you know that they don't. But they do have mechanisms in order to protect themselves. So they have got some chemical and physical protection, just in the same way as animals have physical and chemical barriers to infection and defensive mechanisms to control the spread of diseases once they get in. So the types of plant defences that we've got could be something like passive defences, such as the physical ones, or more active ones, which are chemical. So we have got physical barriers, and these include things like waxy cuticles, cell walls, stomata, callose deposition. Stomata may not seem like physical barriers, but they actually have the ability to close, so of course that does prevent the pathogens getting in. And we've got this material called callose, which we'll come on to a bit later. We've also got chemical barriers, and these chemical barriers include things like nutrient deprivation, so drawing away the materials out of tissues which have been damaged, changes of pH. We've got these things called phytoanticipins and plant defensins, basically chemicals which are being produced in order to kill things like fungi and bacteria. So the first thing a plant's got to be able to do is recognise that it's been infected in the first place. So they do have to detect the pathogen getting in order to be able to mount some kind of response to it. And in order to do that, they've got receptors which are able to detect the pathogen once the pathogen has got into the system. This will then start to signal to nearby cells, and that signalling does what it does in any cell, which is generally to switch on some genes in the nucleus. Now don't forget, when we talk about cell signalling, what we really need to remember is that a signalling molecule circulates, lands on a receptor, and it causes a change inside that cell, and quite often that change is going to involve switching on genes which then produce proteins which carry out activities inside that cell. So things that we could be switching on, we could start to make some chemicals, so some defensive chemicals which might be there to actually destroy the pathogen in some way. And it could also be alerting nearby cells that they need to get ready for other things like, say, fungal infection or bacterial infection getting into their cells. The other thing you can do is you can physically strengthen your cell walls, making it more difficult for the pathogens to penetrate in. So this diagram is really just summarising what we've just talked about. So what we can see at the top of the page there, we've got the pathogen, and the pathogen is producing chemicals. Like we know, they quite often produce chemicals like toxins and so on. And these are going to be detected by the plant cell. So the plant cell itself can actually notice that these toxins are in there, just like they do in animals. And sometimes some of these, when the pathogen tries to get in, what they do is they try to actually digest their way into cells, and from doing that they can actually produce breakdown products of that process, and that can also be detected by the plant cell. So the plant cell can detect the toxins and the breakdown products of an invasion. And so what's going to happen next is that these molecules are going to be picked up by receptors, and that's going to set off a signaling molecule inside the cell, which is then going to go to the nucleus, and the nucleus is going to respond to this in a number of ways. So firstly, it could produce some defensive molecules to directly attack the pathogen. So for example, it could produce an antibacterial chemical or some kind of toxic molecule which should hopefully slow down the pathogen. It can also produce things like polysaccharides such as callose and lignin to try and strengthen the walls and make them less penetrable. And lastly, it can produce more defensive chemicals to alert the cells nearby that they need to get ready and start to produce some chemicals and strengthen their walls. The physical barriers that plants have include things like the waxy cuticle on the surface of a leaf. They also have the ability to close their stomata when gas exchange isn't important, and they also have very thick cell walls, and that obviously gives them strength but also protection for the cytoplasm that's inside the cell surface membrane. Some pathogens have this ability to produce chemicals which can actually digest their way through the cuticle. So these are referred to as cutin degrading enzymes. So things particularly like fungi have this ability. Fungi produce enzymes extracellularly in their natural way of digesting materials, and this also allows them to digest their way inside the plant. Here we've got an example of a fungal hypha which is trying to get through the epidermal cell of something called Arabidopsis, which is a type of plant. And you can see there that it's actually penetrating through the wall and actually breaking into the cell. So how have plants developed this? Now we can see some pictures here of plants. The top one shows caterpillars munching their way through what looks like cabbage. So as plants can't get away from their consumers, that makes them very vulnerable to attack. And so the only way you can actually try and do anything about this is by making the tissues either less easy to access by these physical defences you've mentioned, or slow down the way in which they get into them. So now evergreen plants are particularly good at this. Now if you look there, we've got a picture of holly. Holly is an evergreen, and these leaves are long living. They get through the winters and then they will be there for several years. And although they do shed some of their leaves, they shed them at a very low rate. So these are what we call evergreen plants. The leaves need to be able to stay in good condition for long periods of time. And one of the things that you'll notice about evergreen leaves is that they tend to be very thick, and they quite often have thick waxy cuticles. The leaves tend to be quite rubbery and often have spines and protective mechanisms to reduce the amount of animals that are likely to want to eat them. And if we look at the cactus next to it, they've also taken that even a step further by creating very strong, fierce spines, making it very difficult for animals to actually eat them, particularly as it takes them quite a long time to get to that stage. So they can also not just have physical adaptations, but they can actually reduce their appeal by making themselves taste bad. So quite often plants contain chemicals which we might quite like, but a lot of animals don't like. So for example, the taste of things like caffeine. Caffeine is actually not a very pleasant chemical for lots of insects. So actually, although we use it for making coffee because we like it, lots of animals would find that quite unpalatable. Now, this whole process has developed simply through the process of natural selection. Now, plants which can either defend themselves by having spines or tough leaves, or by producing chemicals which make themselves less palatable, for example, are just going to live longer. And if they live longer, they will pass those genes on which gave them those advantages, and therefore the plants will gradually evolve to defend themselves in those ways. So whenever we ask this question, how does this happen, it's always because those particular organisms had genes which gave them some kind of advantage over the others, so they live longer and pass those genes on. So the successful invasion of plant tissues depends upon the thickness of the cuticle, the presence of a secondary wall, the size of the stomatal pores and the distribution of them, and sometimes also the orientation of leaves. And if we have a quick look at this yucca plant that I've got here on the right, you can see here that it's got quite a thick cuticle. The leaves themselves are quite thick and glossy, and they will actually have also a very thick bark on the stem, so that makes the stems very tough. It allows the plant to get taller, of course, but also provides quite a lot of protection from things like fungi and so on trying to get in through the side. They will have relatively small distribution of stomatal pores, but also the leaves themselves are orientated in such a way that it means that water runs off the surface of the leaf, obviously down the stem to the ground. But it means that, and obviously that's beneficial because it means that the water is going to be picked up by the roots, but it also means that you don't have large amounts of water just sitting on the surface of leaves, which could accumulate large numbers of fungal spores and bacteria. So actually the orientation of the leaves also reduces the potential for microbes to get in. Well, this brings me on to this molecule called callose, which I've mentioned a couple of times. Now, callose is a polysaccharide. It's quite similar to cellulose in that it is made of beta-glucose, but it actually is the bonds between the beta-glucose molecules is different, and you get 1 to 3 glycosidic bonds and 1 to 6 glycosidic bonds, and this creates linear molecules. So what we can see here is we can see the 1 to 3 bonds on the callose, and then it will have some side branches as well, which also are the 1 to 6 glycosidic bonds. And underneath you can see a molecule of cellulose, and cellulose normally only has 1 to 4 glycosidic bonds. So if we just number these bonds here, 1, 2, 3, and then on the other one you've got number 1, so we've got 1 to 3 glycosidic bonds there. And then down on cellulose you can see there's the number 4, so we've got a number 1 to 4 glycosidic bond in the cellulose. So that's just a quick bit of revision really for your biological molecules. So you could, for example, be given in an exam a question where you're asked to compare the structure of callose and cellulose. Although you're not told to learn callose itself, you could be given the diagram and you could be asked to compare it. So it's quite an important molecule to know something about. So cellulose then will only have 1 to 4 beta-glycosidic bonds, and the molecules themselves are alternate. They flipped over, the alternate ones are flipped over. This creates a very straight molecule, and you can see that these bond together to form these fibrils. Whereas callose has got 1 to 3 and 1 to 6 glycosidic bonds, which creates side branches, and it creates a more linear molecule, and therefore that is deposited in a different way. So it doesn't create fibres in the same way. So when plants are attacked by pathogens, they will start to produce lots of callose. And this callose is a physical barrier. And what it's going to do is it's going to be deposited in various areas around the plant cell to reduce the possibility of things getting into that cell anymore. And obviously nearby cells are being alerted to start producing it too. So it could be deposited between the cell walls and the cell membranes so that it thickens the cell walls. It can be deposited within the sieve plates of the phloem so that it's less likely that microbes can be travelling through the plant through the phloem. And also it's deposited in the plasmodesmata between the infected cells. So if you remember, plasmodesmata are these little sort of strands of cytoplasm that extend between one cell and its neighbour, and it allows the movement of materials from one plant cell to the next. And so callose will be deposited to effectively prevent that movement between those two cells and prevent the spread of that disease. Plants produce quite a lot of chemicals, some of which are used in their defence. and they can repel insects, or they can actually try and destroy things as they get into them. And we've learnt about quite a few of these chemicals and extracted them from the plants themselves, and actually used them quite often in our pesticides and fungicides and so on. Here's an interesting example. We've got the castor oil bean, and inside these beans it produces a chemical called ricin. And ricin, you may have heard of, is actually a very toxic poison. Only 0.2 milligrams is needed to kill you. So obviously processing these beans is quite an important skilled task, because it's quite dangerous. What ricin does is it gets inside your cells, and it interferes with the way that protein synthesis occurs, and most things are actually damaged by ricin. Quite a lot of plants produce strong flavours. Herbs and spices have these flavours, which we obviously use for our food. They're often found inside the seeds, and we will harvest those seeds and use them to flavour our foods. A lot of animals don't find them very palatable, so that would actually make them less likely to eat them. A good example here is the capsicum, which you get in chili, and this is a chemical which produces the heat of a chili bean. And obviously there are lots of different varieties of chili, but that's basically because we grow them to try and create different varieties of chili. So quite a lot of people are breeding these very, very strong chilies at the moment, because people want to have very, very hot food. So I have a few examples then. We've got insect repellents, things like citronella from lemongrass. Lots of people use that in things like the outdoor candles, which people put it into the sort of candle wax, and it burns and creates a nice sort of lemony scent, which we like, but insects don't, so it repels them. Pyrethrin is a naturally occurring insecticide which acts as a neurotoxin, and it's particularly found in chrysanthemums, which we have a picture of here. And what's happened in the past is that we've actually harvested these pyrethrins, and now of course we tend to make them chemically, so we don't necessarily get them out of plants, but they have been proven to be very effective. And we also have caffeine, which is toxic to insects. Some people try to put caffeine sprays on their indoor plants to try and reduce things like aphid infestations and so on. And this is a chemical which is found in the coffee beans, which obviously we really like, because we like the bitter taste, and obviously it isn't a toxin to us, but it is a toxin to insects, so it acts as a natural insecticide. Here's a summary of some of the antibacterial and antifungal compounds, including some antibiotics as well. So phenols, which a lot of you do chemistry will be aware of and know that they're quite unpleasant chemicals. They can be used as antiseptics. Glossipol, which is an antibacterial chemical found in cotton. Defensins are a generalised group of plant proteins which interfere with the way that bacterial and fungal cell membranes are formed, so it actually reduces their ability to reproduce. Lysosomes, which you will have actually come across before, these are the organelles which contain enzymes which can break down bacterial cell walls. We often use this in our phagocytes, but it can be found in plants too. Saponins, these are chemicals which occur in many plant cell membranes, which interfere with the fungal cell membranes. They actually have a sort of soapiness to them, which is where the sound of the name comes from. Caffeine, toxic to fungi and insects. And chitinases, these are enzymes which break down the chitin in the fungal cell walls. I know some of you like to prefer to call it chitin, but I would call it chitin. Another group of important chemicals are anti-oomycetes. Now, oomycetes are really similar to fungi, but they are eukaryotic fungi-like microorganisms which have their own distinct groupage. All fungi obviously are eukaryotes, but these are particularly different in that they don't have chitin in their cell walls. But they have a filamentous appearance, and they reproduce sexually and asexually. And we've got probably the most famous example of an oomycete is potato blight. And as you can probably see, the effect that this particular pathogen has upon the potato is quite devastating. The potatoes become completely inedible, the plants themselves die, and you get no crop at all. And of course that was the cause of the potato famine in Ireland, which where lots of millions of people died. So this is a very significant microorganism, and it's quite important that we obviously control it, but plants have their own mechanisms to try and protect themselves against infestations, and particularly these group of enzymes called glucanases, and these ones, glucans are the polymers found in the cell walls instead of chitins, and this is an important enzyme to try and prevent the breakdown of their walls by the enzymes of P. infestans, which is potato blight. Generalized toxins. Cyanide. Cyanide is a compound which occurs in quite a lot of different types of plants. It's toxic to more or less everything, and mainly because it interferes with the way in which energy is converted into ATP in the mitochondria, and cyanide destroys that process. It interferes with the electron transport chain, which you'll hear more about next year. We've got here some elderberries, and these ones are okay if you've cooked them, and people do use them to make wine and so on, but quite a lot of the parts of this plant are actually poisonous, so because they've got quite a lot of cyanide in them. So the other place you might find cyanide, which we're probably all familiar with, is in the apple pips, which has a sort of bitter taste, and that's to do with the cyanide. And again, pips and fruit quite often has a lot of these chemicals in it in order to try and reduce them being destroyed so that they can actually grow. Inhibiting compounds that can be excreted into the internal environment, accumulate in dead cells, or secreted into vacuoles in an inactive form. So it is possible, for example, to put environmental compounds out there which actually inhibit other things from growing. A good example of this is something called alleliopathy, whereby plants release chemicals, trees for example, release chemicals into the ground, and the theory goes, I don't think it's been totally proven, but that they actually inhibit seeds growing nearby, because obviously they don't want the competition. So you often find trees growing with other trees not growing that close by, and that's partly to do with shading, but also to do with the chemicals which can inhibit the growth of germination. But also what we find is that young fruit will often have antifungal or antimicrobial compounds which gradually break down as the fruit ripens, and this means that the unripe fruit is less likely to be affected by pathogens like fungi and bacteria and so on, so that the seed inside can actually mature, ready to be able to germinate. Now, plant defences is a topic which there haven't been many questions on in the exam, so we haven't really been able to find many examples of this for you to practice on. I will have another look and see if I can find you for you to forward. However, what I would suggest you do is a revision task such as making a table or a diagram to summarise the plant defences against pathogens, and we include in things like this the production of defensive chemicals, some examples, you can see the list we've got there, the physical defences and examples of that, such as callose and how that works, and also alerting nearby uninfected cells. Now that then brings us to the end of this presentation on plant defences. So I hope you found it useful, and that then brings us completely to the end on this topic on disease immunity. Thank you very much, everybody. I hope you found these presentations of some help, and that you're now able to get through and have a go at all the questions.